Yeah.
Very good morning to all. Good morning, students. As we are entering our 8th day on the BIIS 12 workshop, we have lectures on microbial biotechnology. Our first lecture is about selection of growth medias for microorganisms. And today we have with us Professor Annama Anil Odane. She is from the DBT ICT Center of Energy Bioscience, Institute of Chemical Technology, Mumbai, a renowned industrial microbiologist. Her area of work is extractive biotransformation, designing and engineering of enzymes, selective isolation and capture of natural bioactive molecules, secondary agriculture and products, process integration and intensification, process development, characterization, and scale-up. Professor Annama did BSc Microbiology and MSc Life Science at the University of Mumbai. Followed up, she did as a PG Diploma in Bioinformatics. After qualifying the CSIR JRF Fellowship and ICMR JRF exams, she entered ICT for a PhD in Applied Chemistry and has been working there since then. Professor Annama has authored and co-authored more than 77 research publications in various national and international journals. She has up to 39 patents, of which 37 is from international and 2 from national. Professor Annama was a part of team led by Professor Aravind Lalli that conceptualized and implemented India's first ever 2G ethanol plant. It was inaugurated in the town of Kashipur in Uttarakhand in April 2016 and is capable of processing 10 tons of agriculture waste in a single day. So with this short introduction, I invite the professor to deliver. The floor is yours.
Thanks for your kind introduction. I'm very thankful for you to invite me for this Biotech Innovation Ignition School. And I am glad that I can impart a little bit of information to the students in this area. So with a short introduction, I will first, the way I have laid down my slides is basically we touch upon why we need to have this kind of an exercise when we are doing any work in biotechnology. And also then we go on to see how the different kinds of media keep changing when we are picking up organisms from the environment, when we are growing them or domesticating them, and finally when we are using them for any production scale. So without losing much time, let me quickly tell you that this area where we use microorganisms for any kind of production or manufacturing basically is referred to as white biotechnology. Here basically what we do is we use microorganisms and their enzymes to manufacture goods, which can be chemicals, plastics, pharmaceuticals, foods, energy carriers, across the spectrum. Any kind of material that you produce using microorganisms or their enzymes come under this white biotechnology. So today, if you look at the general world scenario, people are slowly moving into a bioeconomy framework where we have a lot of engagements with molds, bacteria, plants, and yeasts, so that we can make easily degradable products. Now, it can be in metabolite production, like for example, the washing soaps that you use or the detergents that you use. These are sometimes enzymes from microorganisms, so that your oil stains and other stains that you have on the material is washed off easily. The other sector that we have is waste treatment. If you look around you, there are so many there is so much of municipal waste that is collecting. Recent trends have been that people are using different kinds of waste treatment procedures. It may be algae for waste treatment, or it can be bacteria, which are bacteria and yeast for waste treatment. So you prepare biogas out of it. From yeast, we are using the bio waste and trying and converting it into ethanol, butanol, or any other source that can be mixed very easily. So taking all into these considerations, basically, we first need to look at why microorganisms matter to us and how are we actually slowly moving to use them for different applications. Now, why microorganisms matter? So right from the beginning, we all understand that the origin of life on the earth evolved because of the first organisms to exist on earth are microorganisms, which have come by the fusion of chemicals or whatever sources you may say, and all the chemistry actually drives from there. They are responsible for decomposition, they're responsible for diseases, they are responsible for disease treatments. There are a whole lot of applications and productions that you see. So just to give you a hint of this, basically, if in the morning we have bread toast, right? The bread that is being served is an after product of a microbial activity. In the sense, the dough is made and you leave it the dough either with yeast or without yeast for it to have carbon dioxide generated from the glucose that is there, and then that carbon dioxide basically evolves and the dough becomes bakeable, I would say, right? Now, in that same bread, after it is baked, if you leave it on the table for multiple days without taking proper care of it, or if without consuming it, if it is left on the table, there are microorganisms which will inhabit or inhibit, uh, take control of the yeast, take control of the carbohydrate in that and start to decompose the bread. So you see green molds on the bread. So these are different applications, or these are different sides of microorganisms, how they are involved in the production also, and how they are involved in decomposition as well. So I usually refer to them as very small but pretty mighty organisms, which are responsible for a whole lot of activity that the cell basically does. When we look at microorganisms in a technology, there are multiple aspects of these microorganisms. Many times you are not able to use the microorganisms as such. You need to have multiple changes done to them so that they suit our manufacturing requirements. So primary aspect here is microorganisms do a lot of work by themselves, but when we are looking to domesticate them and use them for manufacturing, it is essential that we look for certain characteristics. First and foremost, we need that it utilizes a whole range of substrates. If it cannot use or cannot digest the food that we are giving it, it will not be very useful for us. Secondly, we are looking at a capability for fast growth and for larger biomass production. Now, by larger biomass production, I mean that if we use a very tiny amount of cells, the conversion that we see may take a lot of time. However, if we change the strategy and use very high concentration of cells, then the entire mechanism changes and then the reactions start to happen faster. Now, these microorganisms are tiny organisms which we cannot see with the naked eye. However, when their cell mass increases, we can actually see them as a powdery material, which is then doing the conversion. So this powdery material can then be used for bioremediation. It can be used for a host of different biomolecules. However, natural strains or native strains may not be able to do certain conversions that we are looking for, or may not have the intensities. Under those conditions, we do genetic modifications so that whichever organism or whichever product we are looking at, we get only that product. One example that I'll give you here is we can use a bacterium called Clostridium for the production of butanol. Now, butanol is required as a solvent in many industries. However, this organism which produces butanol also produces acetone and ethanol along with the butanol. So the ratios that are produced is, if 3 is butanol, then around 2.5, 2 is ethanol, and 1 is acetone. Now the problem is, if these 3 are produced simultaneously, then downstreaming is a problem. Downstreaming means capture and recovery of the material. So we usually do genetic modifications to silence the ones that you don't need, silence the pathways or the reactions that you don't need, and amplify the reactions that you want. So all these things are basically done, or these are modified for microorganisms to function in the technology and to be enabled and to enable large-scale commercial productions. Right. So the applications that we have for microbial techniques are existent across the different areas of life. If you look at historic developments from 4000 to 6000, from 4000 to 6000 BC onwards, we have gradually seen an increase in the use of microorganisms. So right from those days, people have used microbial techniques for different food items. Then we looked at 15th century, we had algae that was being used. Then Pasteur came up, Pasteur and Koch came up in the 19th century. Then we had Edward Buchner again in the 19th century, where he showed that yeast sugars can be converted to alcohol. Then we had large-scale sewage treatment plants in the 20th century. And current day also, we are using a lot of microorganisms for the different applications that you can think of. So just for our convenience, or to enlist, I've just listed out the major areas of biotechnology where we have plant biotechnology, animal biotechnology. Then there is a health segment, health and medicine, and environment segment. The majority of the talk that we will be doing would be around the microbial technology, biotechnology, which has a huge influence on the health medicine as well as the environment biotechnology part. So if you look at the companies around the globe which are leveraging the power, you have all the big names that are there in the enzyme industry, or the manufacture of detergents, or in the manufacture of biochemicals. All the major manufacturers or world leaders use biotechnology, and they are the drivers of the industry. Food people also use a lot of microbiology and industrial biotechnology, like Perry, Marlowe. All these food companies also are drivers in this area, and we see a huge impetus from the market value of 417 in 2018. We're roughly looking at doubling the size of the market. Now, what drives this market? The driving force are basically microorganisms that you isolate from nature. And then after doing a product formation screening experiment, a product yield experiment, then maintaining the cultures, using the cultures in huge bioreactors, all these different steps finally lead us to the recovery of the product and the services. Okay. So therefore, to adopt wide biotechnology, we have a primary requirement that we should develop and isolate robust strains, which will then be used for different purposes. Okay, so how do you do that? Now, if I'm looking at nature, in each and every area, we have different microorganisms, and these microorganisms basically drive all the different activities that nature does. So it be it carbon dioxide capture, or be it the primary production, be it the deposition of materials or elements in the soil, be it respiration, methanogenesis, remineralization. All these different activities are majorly done by the microorganisms around us. So if you pick up any part or any small soil, you can have a very niche microorganism in that specific zone, which can then be cultivated and cultured. So from any of these environments, so from the permafrost or the tundra region or agriculture, lakes, grasslands, wetlands, estuaries, deep sea, surface sea, from each of these sources, you would have a niche organism or set of organisms which are very specific to these areas, which can do their specific jobs. So when we are isolating microorganisms from the nature, we go on to these specific regions and pull out the microorganisms in the form of on these kind of plates or on these kind of microbial media plates, where individual colonies can be picked up, and each of these individual colonies is a population of bacteria which basically exists from the region that you have picked up. Okay, so this plate that you see is it is using a highly concentrated solution of the microorganisms, and slowly by loop dilution method we come to a dilution rate where you can have individual colonies which are growing very smoothly. Okay, so the basic thought process in this entire exercise, which I am depicting by screening of a fungal enzyme, is that you collect the soil or the local fungi on from the environment, and then the soil that you have used, you dilute it with water, you allow the microorganism to come in the water phase, which is then plated out on a plate. And once you plate it down on a plate by dilution, you can obtain the pre-culture, pure culture, and this pure culture is then tested for its feasibility in different environments. So if I'm taking a fungi, for example, from this, I can have different enzymes that are being produced. And depending on what media I am using for selection, I can specifically do a screening experiment or picking up specific enzymes that will be used for the purpose. For example, this is a screening experiment where we have used amylase. Amylase is an enzyme which breaks down starch. So if the fungi is negative, that means it does not produce amylase. It will grow on the plate, but it is not essentially digesting the starch. And if it digests the starch, then to the medium, we add iodine. And if starch is digested, we get this hollow frame where it shows that the starch has completely got hydrolyzed. Similarly, here we have protease experiment. Again, the protease experiment, protease is an enzyme which digests protein. If the organism is able to digest the protein, then it will create this halo around the protein, around the organism, where you can see that there is complete digestion that has happened in this particular area, and therefore you see a halo. So by these methods, we can actually identify a producer or a non-producer and then move ahead from the screening experiment. So what we have essentially done is we have picked up a soil sample or a water sample and then isolated the colony and looked at the different enzymes that colony produces, thereby allowing us to select a select a strain which is a producer of that particular enzyme. Okay, so now let's look at what this media is completely composed of. Okay, now, has everybody had their breakfast in the morning? Yes, no? Or you don't want to reply? Yes. Yes, everybody has had a breakfast in the morning?
Yeah, ma'am.
Yeah. So can somebody tell me what is a balanced diet that we talk about? Any idea of what is a balanced diet that we talk about? Is that any important for human health and well-being?
Like it has
A
the
diet
right
that
Yeah, go on.
ma'am, it must have an adequate proportion of all the nutrients like the fats, the proteins, the fibers. So
Mhm.
it's important for like development of individual parts and individual processes in our body.
Anyone else? Anybody else? What is a balanced diet and why is it essential? I just want to get another perspective. She's not wrong at all. Anybody else? Come on, we are early in the morning. Samra,
it
you were
maintains a balance of nutrients that is necessary in your body.
Yes,
yeah. Samra, you were also saying something.
All the nutrition required for the body to function properly.
Yeah. Okay, so all of you are stressing upon the fact that this is basically nutrients which we have to consume for stable functioning of the body or the well-being of the body. Okay. So let me ask you a question forward. Would a balanced diet be the same for a toddler, for a teenager, for a middle-aged person, and for an old age person?
No, ma'am.
No?
No.
On what basis would you would you decide what is balanced diet for them?
Ma'am, the amount of nutrition and calories required at the age group, specific age group.
Yes. So
As
go on.
no, ma'am. Continue, ma'am.
ma'am, it varies as per age plus even as per the time of the day. Like in the morning, you need to have much more of protein as compared to the fats, as you've already got reserves for it. So it varies, as per the environment plus the time you're exposed to.
Okay. Anyone else? Yeah. So the entire concept that you guys have got it very correctly. So when we decide a diet or when we decide to eat something, we have to pay attention to the components of what we are eating based on a balance in the diet as well as the age and the time that we are having it. Okay. Now, even in microorganisms, we have to keep a very keen eye on what is it that they need for their growth. Okay. Now, our intention is, what is our whole agenda in this exercise? Our intention is basically we want to grow the microorganisms in a very large quantity so that we can produce some metabolite or some product from this entire exercise. Okay. So from this entire exercise. Now, when we are doing this, the intention always has to be that we are affecting a good growth of the organism. So the culture media definition by such is that we are defining food for the microorganism, which is a source of energy. And we also need to put in certain environmental characteristics here so that it can grow efficiently. Okay. So in all these in all these activities, we have to remember one sole thing, that every microorganism is adapted to the habitat from where we are pulling it. And because we want to grow them in the laboratory or grow them in a bioreactor, we have to keep these conditions in mind so that they will grow effectively. So a culture medium is basically a solution wherein all these parameters are taken into consideration. And depending on the type and combination, we have a whole lot of culture medium. Now, why do we need a culture medium? As I said earlier, it is useful for the growth of the microorganism and to provide nutrition such that it mimics the natural habitat. The second part of it would be that we want to study characteristics and properties, and then finally also produce certain metabolites which will be useful for us. So if we want to achieve all these, how do we design a medium? Now, we'd always design a medium looking at what are the microbes' growth requirements. As you mentioned earlier, that we have to take attention of what age the individual is, of what time of the day the individual is eating. All these parameters help us decide what is a balanced diet for a particular individual. Now, having said that, on a similar note, in microorganisms also, we need to pay attention to this. What is the temperature? So if you're isolating a microorganism, or if you have taken a soil sample from the desert, what is going to be the optimal pH or the optimal temperature in that zone? The temperature is going to be higher. It is not going to be 30 degrees Celsius, but on the contrary, it will be 45 to 50 degrees Celsius. So you have to grow the microorganism in that particular requirement. So you have to pay attention to the temperature. We have different kinds of microorganisms which grow in different temperatures. We have to pay attention to the pH. In some of the organisms need alkaline pH, some need acidic pH, some need the pH to be maintained at neutral. So we need to use buffers in the system. So based on all these characteristics, the physical requirement is taken into consideration, and based on that, we do plan the whole exercise. Similarly, the chemical requirement. The chemical requirement, basically the carbon source, the nitrogen source, phosphorus, oxygen. Are we growing complete anaerobic organisms, or are we growing aerobic organisms? All these parameters have to be kept in mind. Now, the next part of it is, what is the role of your media? We'll come to this in a little while, where we are looking at differential medium. But again, is your objective to grow all the organisms in that particular sample? Or are you looking at a specific organism to be grown? Are you interested in just anaerobic organisms? All these choices can be done using a culture medium. Okay. Certain times we also use media specifically designed so that we allow the microorganisms to grow in a highly efficient condition, so that we can map their growth pattern and understand what is their duplication time, what is their growth factor, all these parameters. So in any basic medium, we'll have these multiple components. Major part would be played by water, and then we will have the carbon source, the nitrogen source, the energy sources, mineral salts, as well as specific growth factors such as vitamins and other. Media can be solids, they can be semi-solids, liquid, and based on the ingredient, many a times you will have a synthetic medium. Synthetic medium will be where everything is per se defined, right? And each and every component going into that, the user will know about it. In a complex medium, you basically have some kind of powders going in, but you actually cannot pinpoint that, okay, the carbon is X amount, the nitrogen is X. These are complex mediums. And then you have special mediums which are required for the growth of organisms. Now, first and foremost, let's look at the definitions between defined and undefined. In defined, what we usually do is there is a defined source, as the name suggests, defined source of carbon and nitrogen, glucose, glycerol, all these components, along with the ammonium salts and nitrates, these are all organic components which will go into the defined medium. Okay. The next comes the complex ingredients. This consists of a mixture of many chemical species in unknown quantities, maybe yeast extractant, casein, all these different kinds of materials will go into this. So if you're looking at examples, I will give you an example of a chemical medium. So look here, you have glucose, NH4H2PO4, the phosphate moiety is also defined, the NaCl is defined, the MgSO4, K2HPO4. So as I said, every chemical that goes into the media will be very clearly defined. On the contrary, we have complex medium, and these complex media will be like peptone. It's a nitrogen source, but it may have vitamins also. It will beef extract. Again, it's a complex source. It will have all the small ingredients that are possible, NaCl, agar, and water. So here what you see in complex media is we don't really know what content or what chemical nature is there in this material. Okay, and therefore it is referred to as a complex or undefined medium. What we next come to is enrichment medium. Now, special media. So in special media, there are multiple types of special media, some of which are enrichment medium. So what these will do is they will enrich only a single organism. In comparison, they will silence all the rest of the bacterial population. Just one single microorganism will be enlightened. So this is usually done. So when we have, I'm going to give you an example. If we need glucose assimilating organisms, or we need some other source. So you have a vegetable mix, and in that vegetable mix, there is a C5 carbon sugar, and that C5 carbon sugar needs to be degraded by microorganisms. So you basically provide that C5 sugar, and you will do a enrichment for only the microorganisms that will grow in that condition. Suppose you have a soil sample and you want to understand only microorganisms that grow at higher temperature. So you will leave that plate at higher temperature and you will add media components in such a way that there is very low evaporation at higher temperature, and you can incubate it for a longer time. That again will come under enrichment medium, where you are increasing the number of desired organisms. Okay. In selective media, on the contrary, we will encourage desired organisms and suppress unwanted organisms. Now, enrichment mediums are usually liquid, whereas selective media would be solid media, where you can see the difference or see the colonies that are growing. They have a very thin line of difference though. Next that comes is the differential medium. In differential medium, we are able to distinguish colonies of a specific microorganism from the other. Another type of media are anaerobic media, where the growth of obligate anaerobes happens in this condition. The next that we look at is transport medium. Now, this transport medium was used very routinely in COVID cases, where COVID times, where the samples from individuals needed to be transported to the RT-PCR facility for analysis. So they used to take swabs and put it in specific medium, which would then preserve the microorganisms or the virus in that media till it is being analyzed. So essentially, transport media, as the word suggests, are media which are solutions of buffers, carbohydrates, peptones, and nutrients, which basically are desired to preserve the viability of the bacteria during that transport system. Let's look a little bit more on enrichment media and the different media that we have discussed so far. So enrichment media will allow the growth of only one particular microorganism. We usually have inhibitors which are added to stop the growth of unwanted, usually in liquid phase, as I told you. They are designed in a manner that all unwanted commensals and contaminating bacteria are repressed here. Some of the inhibitors that are added to the enrichment can be antibiotics, dyes, chemicals, pH alteration systems, etc. Now, examples of these will be Lowenstein-Jensen medium, selenite F broth, tetrathianate broth, alkaline peptone water, pseudo-celled agar. These are enrichment media. Selective media would be wherein you are using this only for the growth of selected microorganisms. And the selection is by adding antibiotics which prevent the growth of the other cells lacking amino acids, and these may also contain stains and other color indicators that can be used effectively. Differential medium. Now, differential media basically distinguish one organism from the other. The shape, color, growth pattern, all these are observed in differential medium. So dyes in the medium, like pH indicators, indicate the differences between organisms. Examples would be eosin methylene blue agar, McConkey agar, mannitol salt agar. All these are differential media that you have. Now, based on what microorganism you need to use, we also define the media that can be used. So we have here again a selection or an enrichment medium wherein if you want to grow bacteria, you know for sure that okay, this bacteria are existing. So then for isolating those bacteria, you use specific media which will not allow the growth of yeast and fungi or actinomycetes. Similarly, if you want to grow yeast and fungi, we have a different set of media that will enable the growth. And for ice and actinomycetes, again, we have specific media that will not allow yeast and bacteria to grow, but only allow actinomycetes to grow. Okay. Now, one of the examples of such a screening exercise, which we had done long back in 2017, is something that I show here. So we had picked up seaweeds from the shores of Aberystwyth, and then these were fresh seaweeds as well as rotting seaweeds. And then with this entire sample set, we did a whole series of isolations. And we isolated around 367 colonies from fresh ulva as well as 482 from the rotting samples. And these samples were isolated and put onto different plates, which are basically differential plates with different enzymes, which show different enzyme properties. So this was a plate where we're looking at ulvandiases, this was beta-glucosidases, exocellulosases. And on these plates, on individual of these plates, 1,000 odd colonies are pinpointed, or they are put in dots. So each dot that you see here is a colony. And if you see zones of clearance around those colonies, then those are positive strains that we have taken up for analysis. And post this, we can then send for a 16S RNA gene sequencing to identify which organism is basically present, which is the organism that is present there, so that we can identify the gene of itself. So the workflow for basically generating these kind of colonies is that you pick up a soil sample or any kind of sample, do the cultural isolations using specific media. The choice of media, the way you do this is very, very crucial in the final product that you get or the final clone that you get. Now, once you get the clone, then you check for its enzyme activity or for the property that you are looking for. If it is in good content, then you do a protein isolation and go further on. But if it is not, then you go back and do a sequencing and generate mutants. Again, you use media for selecting those better mutants, best performing mutants, which you analyze using the assays later on. So for any exercise that you do in industrial biotechnology, these selection medium that you use is very, very crucial. And these lay the foundation to what kind of colonies then. Usually when people do these kind of experiments, many a times they end up with either a bacillus or a serratia, or, which are common flora microorganisms. Always remember that any mistake that you do in your culture isolation or your contamination, your technique for doing this would eventually result in in an organism from your environment being a populant of the plates or the selection plates that you have. And therefore, this screening experiment has to be done very, very carefully, so with equal amount of repeats, so that your selection is very uniform and correct. Okay. Once we have selected, we can go down to enzyme isolations and multiple other things. I will not spend time here. So that was the story about how you isolate microorganisms from the environment. That is that you dig into a soil or a water sample and then pick up organisms that are performing under those conditions. But that's not the end of the story. That's just the beginning of the story. Now, once we have got the organism, what next? The story starts there, because our aim is to manufacture a product from this particular microorganism. And that is why we have invested so much time and effort to actually isolate the organism from there. Okay. So once we have isolated, there are different things that we need to do with it. Growth, you need to just grow them. So for example, the dairy industry needs a lot of rennin, and for the cheese manufacturing, they need a lot of proteases that are there. Okay. Now, proteases will come from bacilli or any other microbial source which is producing these proteases, or from plant sources like papaya, pineapple. Any of these sources can also give you proteases. But point is here that if you're isolating, or if you are using a microorganism to produce these proteases, you need those microorganisms in tons and tons of quantity, so that you can extract the enzyme and then give it to the industry for their manufacture or for the use. Similarly, for the detergent industry, you need enzymes, you have to make in high quantity. The baking industry needs the organism per se, it needs yeast. So you need tons of yeast, dry yeast, that are made every day to fuel the baking industry. So what that essentially means is we need systems where we can grow the organism that you have isolated. Okay. Growth is one part, where growing contains low amounts of nutrients, and it creates the raw material for further fermentation processes. And next is fermentation. Fermentation media would mean that you have a specific final product in mind. For example, alcohol in mind. So alcohol comes from glucose. So once the organisms are grown, they are then transferred to a fermentation media, where the glucose is then converted to ethanol. And this ethanol is then used for different applications, either for mixing with petrol, or for drinking purposes, or for chemical industry purposes. There are a whole lot of applications around ethanol, methanol, and other molecules. Okay. So the difference between these two systems would be that in a growth medium, I will just give around 1% of glucose, because I'm interested on only the cells growing, the cells going through a binary fission exercise or a vegetative multiplication exercise, and increasing their number so that we can use them for the fermentation system. Now, with the fermentation system, for fermentation of or production of alcohol, we need to have almost like 12 to 13, because this is what comes from the sugarcane juice, 13% of glucose that is there in the fermentation medium, so that it can effectively convert it into alcohol, which can then be distilled out and purified and taken further for the different applications. Okay. So when we are designing these, we need to be very careful about that. The nutritional requirements also need to be met, but we have to get maximum yield, minimum yield of the undesired product. You have to have it in a consistent availability throughout the year, and it should be very cheap as well. So when we are designing media for manufacturing purposes or the fermentation purposes, we basically put up a balance of the carbon source, the nitrogen, the energy, the oxygen, nutrients, all of which are going into the biomass, the product, carbon dioxide, and water and heat. So an input output map is usually made so that we know how much of product can be got from the ingredients we are adding in. What kind of differentiation would go into the biomass buildup, and what kind of carbon will go into product. At the end of the day, what we are attempting here is basically to convert carbon from one form to the other so that it can be used for viable purposes. So usually the elemental composition of the microorganism is taken into as a guide, and based on the chemical pathways that the microorganism does, we design these media, specific media components to give us the products that are usually there. So usually when we are doing a fermentation medium designing, we prepare charts and we do a whole lot of permutations, combinations before we actually come down to optimizing the media. Okay, we start with raw medium from the literature. Then you select your production medium, which is usually a complex undefined media. And then once we have designed this and understood what is the output pattern, we go back and do a database preparation for individual component screening and understand the significant resources that are going into the system, which post that is optimized using design of experiments and a series of statistical designs, which allow us to identify the core factor and finally predefine the medium, which gives us maximum output from the system. Now, using these methodologies, we basically underline what we need as a fermentation medium, which is very crucial. Now, in the fermentation medium, our focus is that we get our product in high quantity. So what we want from this entire exercise is we want that our microorganisms are happy. They have luxurious metabolism in the sense that the entire pathways that fuel the production of the product are happening in a very consistent manner. The fermentation the organisms are hyperactive because you have a whole lot of nutrients, and it results in decomposition, consumption of the excess nutrients, and therefore the fermentation product. If we do not give in high quantities, the organism will use up the nutrients just for its growth. It's never going to produce that excess amount of product that you are interested in, or the output product that you're interested in. Again, here we have synthetic medium as well as crude medium. The definitions are almost same as defined and undefined medium, right? It is easier to go with crude medium because it is always there, it is cheap. But when we want to make crucial or batches go wrong, it will be difficult to understand because of what things went wrong. And therefore, people usually insist on initial phases. If you're looking at a pharma industry or very specific product requirements are there, then we would want to go with a synthetic medium, which can be designed and redesigned to give us the product of interest. So advantages and disadvantages of synthetic medium. Here, the advantage would be that it lacks sources of protein and there is no foam formation. Product recovery is easy because you have pure components, but there are a lot of disadvantages. Being cost of medium, the fermentation becomes uneconomic and non-profitable. It never is used at an industrial scale because it's very expensive. This is for small scale or for applications where you need a lot of fine tuning. In the crude medium, on the other hand, we have inorganic salts go in, which basically do the act of neutralization. It does the act of balancing, also of pH maintenance in the system. Carbon sources here you can have from simple to complex. Simple would mean that you use starch, you use cellulose, use glycerol, all these sources as your carbon source. And the selection of the carbon source always depicts the cost of the raw material. And then in most of the fermentations, a crude source of carbon is preferred because then it becomes a viable option for doing the fermentation. Next comes the nitrogen sources. Here salts of urea, ammonia, nitrate can be used, and the fermentations are non-proteolytic in nature. You can use urea, ammonium, and nitrate, but when you are proteolytic, you basically have animal or plant raw materials which are used in this process. Okay, you have growth factors being added, precursors that may be part of the system as crude components. So for example, in vitamin B12 production, we need cobalt chloride in less than 10 ppm on the fermenter. Now, multiple of the products that are of the nitrogen source or the carbon source would have this essential amount of cobalt chloride in it. And therefore, it is not always essential to add these precursor molecules. For the buffering, you have proteins, peptides, and amino acids, which act as good buffers during the system. So when we are doing this media optimization, you have to consider biomass growth phase in isolation, and only then basically we look at efficient growth and further product formation. So the classical design in which people work is you change one variable at a time. So total number of variables would be X into total number of variables would be N, and experiments that you would perform is you would perform different levels of optimization. So first you would optimize carbon, then you would go down to nitrogen, then the buffering, all these different levels. If you have to optimize one by one, then you have a host number of experiments, which would be X multiplied by X raised to N number of experiments you would have to be put up. Now, such experiment statistical tools are available for this designing, such as Plackett-Berman design and multiple other design, which ease the whole thing. Also, automation has stepped into the enzyme media designing. Only 0.1 to 1% of the estimated number of microbial species would be isolated and successfully developed. So, based on the knowledge base that we have, people have created a media database where almost all the growth conditions are defined. And over the years, whatever literature has been developed for organisms and specific genome sequences, that is basically put up in this, and we grow it on a complete mode. So in here, once we put the name of the organism and multiple other parameters, we intend to extract information from the growth data, and with the basic understanding of media components and compounds that are involved in the growth pattern, we can basically make a prediction of how much of biomass can be yielded from the whole thing. And also basic components or compounds, what would be the amount that can be coming out from this particular media if you're using for a particular strain or organism. So here, this automated screening methodology brings together literature sources from a wide variety of work that has been done across the globe, and it contains chemically defined media, because that is where you can have absolute control, and the product that is formed can be predicted based on the metabolic pathways. There are links to other compounds from other databases, and this focuses on organisms with existing in silico models and allows the organism specific media conditions to improvise, and you can have more specific organism specific media. And this includes only species which are highly sequenced so that you can gather this information. People are also using artificial intelligence in the media optimization. And the aim of all of these is to yield the metabolite, highest amount of metabolite that is produced. So in total, if you're looking at media optimization strategies, the techniques that you're using is very important. You could use traditional one-factor techniques wherein either the removal or supplementation or replacement of any important component of the media would help us decide whether it is going in the positive direction or the negative direction. Or we can resort to advanced statistical or mathematical tools like ANN, RSM, or the Plackett-Berman design. And these can be used for screening a range of compounds and combinations and interactions to allow us to come to the optimum media design. There are a lot of bottlenecks and problems associated. Primary one is labor cost, and these are open-ended experiments. And when we're doing experiments in the shake flask, they may not exactly match with the fermenter scale studies that we are doing. And this creates a huge gap in what is needed and what is actually done on the desk, because not everybody will have fermentation capabilities to take it to the larger scale. And it's also assumed that best media contained in the flask will be the best at the fermenter. But this is not the case in many of the studies, because in the fermenter, when you are using agitators or air bubbling, there is foaming, there are multiple, the cells may not be able to handle the shear. So this may not be the exact methodology to do it. But however, for ease of convenience and economic constraints, we basically stick to this and make this assumption that whatever you get in the shake flask will perform in a similar way in the fermenter too. Now, when we are designing the media, we have to remember a couple of things, that these microorganisms as cells are dynamic in nature and they have a lot of internal control systems. When we design, we basically assume them as black boxes and do the optimization design. And therefore, most of the times we end up in an arbitrary value, which may not be the final endpoint. And it is designing is like a continuous process that we usually go on. And many a times we look at new components or media to increase the yield so that the entire process is more fruitful. Further directions in this exercise: the evolution of media formulations through screening. These can significantly improve the growth, viability, and overall yield of the product during the process phase. So this exercise needs to be done where we are putting our strains, the mutated ones as well as the native strains, through this exercise of optimization so that we can come to the best value that is available. There are still points that need to be considered for precision and further optimizations. And these specific points need to be focused around the productivity of the method or productivity of the product that you are looking at. How much can the microbe produce? So all the optimization that you have done is with respect to a particular microorganism with a particular product in mind. Without these two combinations, the entire exercise becomes futile. Most of the organisms which we are growing in present conditions may not be able to use raw material. And therefore, through mutation, we need to look at assimilation of low cost substrates, which will reduce the overall cost of fermentation and allow us to have more affordable products in this entire exercise. So that's all from my part. Thanks a lot for giving me this opportunity. And now we can have questions if you have any.
Thank you, Professor. Students, the session is open for discussion. If you have any questions, you can ask.
Ma'am?
Yeah.
What exactly did you mean when you said that microbes are treated like a black box without considering their internal controls?
See, usually when we have, suppose when we are growing the organism on an undefined medium, right? We basically do not go into the minutest elements that are there. Let me give you an example. If you are taking taking whey, dairy whey for fermentation, we know that there is lactose in there, and the organisms that can ferment the lactose are the ones that we are picking up for the process, right? Now, are we good enough with that lactose to trigger the growth of lactobacilli on a tangential mode or in the log phase? Do we need to supplement a little bit of glucose so that it grows to a certain extent, post which the lactose can be utilized? Now, all these questions come when we have a very detailed understanding of the metabolic pathways of the organism that you're considering. Right. At the moment, not many organisms have their entire metabolic pathways get used. So we usually treat them as a black box, wherein we feed. We know that, okay, this organism uses lactose. So we feed it with lactose, and then we wait for the lactic acid product to come in. The minute changes that are happening in the cell metabolism are things that we are not mapping, because every media change would cause changes in the metabolic profiles, which will directly or indirectly affect the end product. So what I meant by treating as a black box was with regards to this aspect, where any designing or any execution at the moment is not taken into consideration, is not considering the complete metabolic map of the said organism.
Okay, got it. Thank you.
Yeah.
Any other questions?
Hello.
Yeah.
Good morning, ma'am.
Yes.
First of all, thank you for the lecture. Ma'am, so I have this question about the experiment you guys performed in 2017. You show that in your slides. So I just wanted to ask, like, why did you choose that particular plant? Because it's not like you knew that there was this particular enzyme in that plant. How did you come up with the idea of selecting that particular plant?
See, we had a project at that point of time where we wanted to valorize the seaweed, because the seaweed then question was one that causes blooms across the European oceans or beaches. And there is a lot of this particular seaweed which flows on to the beach. And because it is a sulfate containing weed, it causes a lot of H2S smell on the beaches, because of which you cannot go on. So there was an entire project that was funded for the particular with the particular aim of evaluating how you could convert this seaweed into value-added products. As a first part of the exercise, what we looked at was to actually understand what kind of degrading enzyme organisms grow on the surface of these seaweeds, so that we could isolate enzymes which would efficiently break down the polymer or break down the seaweed to get derivatives or products out from there.
Okay.
So that was the thought process.
Okay, thank you.
Yeah.
Also, do you mind sharing the PPT with us?
Yeah, I will share.
Okay, thank you.
Any other questions? Anyone else?
Ma'am, one more question I have.
Yeah.
Yeah, so I mean, if you have a problem statement like, the banana pandemic, I am sure you're familiar with it. So I was wondering, how do you approach these sorts of problems? Should you go with the microbiology side, or you should change the banana plants in such a way that they are not, uh, they are not readily invaded by the fungus, or you should, how do you approach solving these kinds of problems? What?
See, usually these problems have multi-facets. So today, if you're going onto the farm and telling the farmers to change their crop, is it going to be implemented? You will face a huge resistance, right? Okay.
Right.
So every problem has a multifaceted approach wherein we look at short-term goals, medium-term goals, and long-term goals. So in the short-term goal, basically, it is something that you can implement quickly so as to give relief. In the midterm goals is wherein you initiate a study with certain set parameters where you can show that if there is a change in the plant variety, you may get a better response. And once you finish that stage, then comes the final stage, the long-term goal of removing the entire crop variant and introducing a new variant on. Right? So most of the problems that we have, or all the problems that we have should be sorted out in this kind of a stepwise manner, where approaches for specific periods can be defined, and they then lead to proper solutions. Otherwise, if you go and, if there is a wall to be broken, tomorrow you get a bulldozer there, the wall can be broken, but what do you do after the wall is broken?
Right.
You need to have solutions to the problem. You can tell the farmer, boss, your crop is a non-resistant variety. You need to change it. But there are economic, mental, financial implications to whatever you say.
Mm-hmm.
And
Right.
therefore, most of these problems need to be sorted out in a gradual, stepwise manner, addressing immediate need and long-term need.
Okay, ma'am.
Right?
Right. Thank you.
Anyone else? No more questions. Then I would like to thank Professor Annamma for joining us today. Thank you so much, Professor,
My pleasure.
for the valuable time. Thank you.
Thanks everyone for listening carefully and for the opportunity. Bye.